Hello, everyone. Welcome to the Capstone project's presentation by one of our star students from the Foundations course, Stan Siranovich. He'll be talking about predicting tertiary protein structure using machine learning. All right, take it away, Stan.
Okay, thank you, Raj. Well, the title of my presentation is Big Molecules Meet Big Data, or Predicting Protein Tertiary Structure from Combinatorial Chemistry Data. Now, that sounds a little bit complicated, but it's not. Basically, what we're going to do is use 9 factors to predict one outcome variable, the RMSD, which I'll define in a couple minutes. And along the way, hopefully, we'll be able to gather enough information so that we can make some inferences from the chemical properties to the physical structure of our final polymer. Okay, let's start with an overview and a path I'm going to take through the analysis. First, we'll start with the project description, which I just did. From there, we're going to go on to a discussion about domain knowledge. In some areas, domain knowledge is a bit more important than others, and this is one of those cases. So we're going to deal with some combinatorial methods in chemistry, which is used to collect the data, and then give a very, very brief discussion of protein structure and protein folding mechanisms. Then we're going to take a look at our data set, do an exploratory analysis of our data, which will make up the bulk of this presentation, do a little bit of model building, specifically some regression models, and finally move on to our results and discussion. Okay. Project description. What we're going to do here is predict a protein tertiary structure. What we're going to use is the Root Mean Square Deviation, or RMSD. And what this is basically is a measure of the distance between equivalent atoms of a very complex model of our protein and actual protein itself. It sounds a bit complicated, but you can think of it as just basically predicted versus actual. And that's what I did throughout the course of this presentation. So we have a predicted number and an actual number, and we're going to see how close they are. Now I mentioned domain knowledge. We're going to use some combinatorial chemistry, protein structure, and finally, since this is a real-world situation, we're going to talk about a real-world consequence that happens when a protein folding goes wrong. So, combinatorial chemistry, it's a multi-step, very highly automated chemical synthesis, analysis, and data collection. It makes very heavy use of robotics and the automated data collection, and it's a relatively recent development. In fact, it's only come to the fore. It's only been made possible in about the last 20 years or so. It's obviously got some academic potential, but it's got commercial potential also, mainly in the field of drug discoveries. All the big pharmaceutical companies are using these sorts of analyses. And none of this would have been possible without our big data methodology. That is, the ability to collect and store and manipulate and profitably look at the data. Now we come up to protein structure. Okay, protein is a specific sequence of amino acids. Basically, what it is a polymer. It's a biological polymer, and the monomeric units are the amino acids. And as they are reacting with one another and building this long chain end to end, they go through some intra and intermolecular interactions and form a 3D structure. And that is what we're interested in. So when we talk about protein structure, we talk about 4 areas. The primary structure, which is a linear sequence I just mentioned. The secondary structure, which is the 3-dimensional arrangement. And the the secondary structure is local three-dimensional arrangement of the backbone atoms in this long polymer chain. Tertiary structure that we are interested in is how this long chain-like molecule folds itself in three-dimensional space. And finally, the quaternary structure. We're not so much interested in that, but for most proteins, that is the end result. And the quaternary structure is when two or more of these protein molecules come together to form larger complexes that have biological activities. And here's what it looks like. It's just a rough illustration, but it gives you an idea of what we're talking about. You have this long chain forming from the amino acids, and they can form sheets or sometimes spirals. This happens to be what's known as an alpha spiral. And it folds itself into some convoluted shape. For those of you who study these sorts of things, this is the low energy state, the lowest potential energy. And several of these can agglomerate together to form our biologically active polymer. Now, here is a real-life example. There's a lot of research going on right now regarding Alzheimer's disease and a beta amyloid protein forming plaques and precipitating out of solution, et cetera, et cetera. But one real serious consequence of protein misfolding is sickle cell anemia. And here what we have is an amino acid valine being substituted for another amino acid, glutamic acid. This isopropyl group here on valine is hydrophobic. It doesn't like a water environment. It tends to want to put itself into the interior of this structure, whereas a glutamic acid is hydrophilic and wants to be on the outside. So you can see that just the substitution of this for this could have very, very serious repercussions. Now, root mean square deviation. What do we mean by that? Let's look at the bottom picture here first. This is a representation of what we just looked at, our long protein molecule. And the blue one is the predicted conformation in three-dimensional space, and the green is the actual. And what we want to do is measure the distance between these little dots here. Let me try and expand that a little bit so you can see it better. The distance between the dots. And the way that's done is to measure the RMSD. Basically what we do is measure the difference for each corresponding atom starting at one end and going to the other, of both the predicted and the actual. Find the distances, add them up, divide by the number, and take, distances squared, add them up, divide by the number, and then take the square root, and that gives us the RMSD. And you can see that the lower the RMSD, the lower the two conformations, or the closer the two conformations are going to be. So ideally, we'd want that to be equal to 0 or very close to 0, because that means we have a good prediction. Now, moving on to the data set. This is the physicochemical properties of protein tertiary structure data. It was originally published here about 2002, 2004 or so, and I downloaded it from our good friends, University of California, Irvine's Machine Learning Repository. A couple things to note. Number one, there was absolutely no documentation with this thing. And number two, the exposed area we'll get to in a minute. Move to the next slide. And this is an overall description of the data set. What we have here is laboratory data. It's nice and clean and tidy. And we have a table of factors. As I mentioned previously, there's 9 factors. And there they are. And what do we have here? Well, we have total surface area, nonpolar exposed area, fractional area of exposed nonpolar residue, et cetera, et cetera. you can see in about half or maybe a little bit more of these, we're talking about a surface area and a nonpolar residue. So I wonder if they could be different measurements of the same thing. So let's start the exploratory analysis. What we have here is a lattice plot. Oh, I forgot to mention in the data set there are 45,730 records. So this is an excerpt from the lattice plot. We simply used the plot function, and I plotted a sample. Even when we used the alpha function, there was just way too many points to have anything intelligible come out of it. Also, we have our predicted outcome, the RMSD, plus 9 predictor variables or factors. So that's a 10 by 10 matrix, too much to fit on a screen. So this is the upper left-hand corner of that matrix. Well, what do we see? It looks like we got some serious correlations going on here. If you start reading down the column from F1, we see we have some strong correlation with F2, and not so much with F3 maybe, but we've got a real strong correlation with F4 and F5. So that's one thing to note. The other thing to note is right here with the RMSD, is we look at the plots of RMSD versus our factors across the top, we see a kind of an unusual distribution here. Looks like it's bifurcated somehow. We have two groups. We have this group here, which seems to go up as F1 goes up, and then we have this one here that seems to give us about the same RMSD no matter what F1 is. Same with F2, and it's kind of blurry here on F3. We see the same thing with F4 and F5. So we note that. So let's take a look at the correlation. That's on the next slide. That is right here. And of course, this is mirrored along the autocorrelation where it's 1. So bottom half is the same as the top half. But let's look here at the bottom half. What do we see? Well, let's look at F1. Oh, correlation with F2 over here looks pretty high, 0.91. And we drop down with F3, 0.1393. Here's 1.9. They're all quite high. So that's one thing to note. So that's going to present somewhat of a problem for our analysis because they're all correlated with one another, which says, well, how much data do we really have that we can bring into play with the analysis? The other thing to note is the correlation of correlation coefficients of the RMSD with the various predictor variables, our factors, they're quite low, unfortunately. Highest one here is with F3 at 0.37, so we take note of that and some of the other larger numbers, F2, 0.16, and we're going to use that in our further analysis. Now, this is a plot of RMSD versus total surface area. Again, I took a sample size of 1,000, and we can see this bifurcation really clearly here. We have this group, and we have this group. Looks like we may have some non-linearity here. So we'll take note of that and move further in our exploratory analysis. And what I like to do is, before I do anything, basically the data set, I like to look at the descriptive statistics for the data set. And here I just use the describe function from the psych package, and then I use the select function. I think that was from dplyr or maybe tidyr, I can't remember which one, but I selected what I wanted out of the describe output. And what I selected was mean, standard deviation, skew, kurtosis, median, and range. I probably could have left the range out. But first thing I do is look at the means and standard deviation. I don't really see anything there. You can divide standard deviation into the mean once, twice, 3, 4 times, which is fine. Tells us nothing really strange there. But if we go further over here to the right and we look at F7, we see a skew of 20, almost 21, and whoa, kurtosis of 804. Well, what's that tell us? That tells us that it's highly peaked, doesn't have much of a shoulder, and there's probably something lurking far out into the tails. So let's take a look at that. And here what we did was just simply plotted a histogram and data. Now here is the entire data set, all 4,730 records, and we can see it's highly peaked. We have no shoulders out here, but there's something going on because our axis goes all the way out to, looks like 100,000, is that 10,000? Let me make that a little bigger. And here we took another sample since our data set is so large, and yes, we see it's highly peaked, and we have some outliers way out here, another one way out here, and way out here, but notice the y-axis is a percent of the total. So most of it is highly peaked, but it doesn't look too unusual, so we'll take note of that, and we'll just move on for our regression analysis. Now, keep in mind that this regression, we're still in the exploratory range of our analysis. And usually what we do here is say, okay, separate it into a training data set and into a test data set. But remember, we found all these anomalies. So let's not quite do that yet. Plus, I have some other outside information that I'm going to bring in. But moving on to analysis, we just used a simple LM model. F3 had our highest correlation with the RMSD, so let's bring that in first and look at the adjusted R square. It's only 0.14. By the way, all these are highly significant at the 4 decimal places, but I just didn't see adding another column. So let's add another one, F3, which was the second highest correlation. It brings us up to 0.17 and so on and so forth till we get up to an R squared of about 0.26 or so. These are all rounded. And it looks like we hit a spot where we're not improving any. And since these analyses are so simple and easy to do in R squared, just for general interest, we took a square of F2, did nothing for us as far as the adjusted R square goes. Took a logarithmic function of F3, did nothing. And we looked at an interaction term here, F4 plus F2 plus F4 cross F2. We got the same 0.26. So the best out of all these, keeping in mind this is still an exploratory analysis, looks like it's RMSD5. Because we got the 0.26 R squared, and we only had 2 factors, so it's nice and simple. And here's what the regression looks like. And maybe if we have effects something like this going on, that explains our bifurcated data set. If F4 were real large, then that would tend to decrease the Y. And if F2 were relatively large, that would tend to increase it. So we have 2 competing factors here, one plus, one negative. Now this is just a copy of one of the previous slides. It's our data set slides. And notice that the factors which added something to our R squared were all some function of the area. F3 was our largest, and that was the fractional area of the exposed nonpolar residue hanging there off of the linear protein chain, F4, F5. They're all something of exposed areas. So, results and discussion. What can we learn from this? Well, first of all, the exposed nonpolar surface area seems to be what's really significant here. Our highest R-square is 0.26, and that was achieved by 4 models. And again, it was with our factors F2, F3, and F4. They were all our old friend here, some manifestation or some calculation based on a nonpolar surface area of some sort. So that's obviously important. For those of you who deal in the chemical sciences, and you're thinking, gee, these are rather minor effects. What about van der Waals, attraction, and this and that? And the other thing, maybe some disulfide bonds. Well, all those were used up in the secondary structure, which we don't try to predict here. And once again, our chosen model was the simplest one. It gave us the highest adjusted R squared, and the interaction term was really of no value. And the evaluation of our results, I think, depends on somewhat on the context here. The world I came from, industrial chemistry, 0.26, that would have been fairly poor. But what we're dealing with here is a complex protein structure. There's probably 22 or 24 different amino acids that come into play. And we're talking about rather rigid structures also. And it's a real complicated situation. Plus, we don't have a whole lot to work with because our factors, our predictive factors, are correlated in many cases. So let's move on to future work. Future work, investigate how the factors were derived. There may be some hidden factors in there which were very important, which were somehow confounded with all the calculations. Investigate the PDB data stores. These are all available online. I won't go into detail there. Another thing we can do is search literature for RMSD and other single point predictions to see what's out there and see what other people have done with this before we launch our analysis. And finally, investigate the underlying nature of the bifurcation of the RMSD plots using classification or cluster methods. The interesting thing about this is the fellow who posted the data set, P.S. Rana, did his original study, and he found that he was able to explain well in excess of 80% of the variation from the mean, but he did not use regression. He used clustering analysis, and that's one thing that I started to do, but it's not ready to present yet. And besides that, I am out of time. So that pretty much wraps it up. I have some review slides if anybody would be interested in downloading them. And I've made this whole presentation available under the Creative Commons 3 license. I think it is share and share alike, non-commercial. All I ask is some attribution. And that's it.
Thank you, Stan. That's a really fascinating project. All right. Time for questions. Feel free to ask questions in the chat box or unmute yourself and ask a question. Come on, I know you have questions.
There got to be some questions.
All right, actually, well, I have one, so let me start that. I'm really fascinated by that bifurcation thing.
Yes.
How do you think you can use that to get a better prediction?
Okay. Well, first of all, separate it. Let's go back to the slide.
Yeah.
Right there. Maybe let me make it a little bit bigger. Oh, I'm glad you asked that because it brought up something that I didn't mention. First of all, the bifurcation looks to be about in here. There's like a...
It looks almost like there's a straight line that can separate the two classes.
Absolutely. Absolutely. So I'm thinking separate it into 2 separate groups, and I already started doing that. I ran an H-class on it.
Yeah.
Okay, and
cool.
if you run the RMSD, depending the variable that you feed into the H-cluster function, you can either get, this is the other strange thing about it, you either get, say for example, you run a t-apply mean on the result from the H-cluster. In some cases, like with the RMSD, you get hugely different averages for the mean. And in other cases, they're quite close. So that tells me that some of these factors here are more important than others, and maybe they should be weighted somehow. The other thing is, the regression you run on this cluster is obviously going to be different than what you run on this cluster.
Exactly.
Some nonlinearity here.
Yeah.
Okay, so maybe this would be something that we could use the carrot on.
Yeah.
And I thank my advisor for suggesting that one. For that carrot is thank you, Matt, if you're listening, for classification and regression testing. That's what the carrot test does for
a nonlinear method will work really well. I mean, those are a couple of things I can think of. One is a nonlinear method of regression where it looks not only like they're two separate classes, but they can be captured by a very nice kind of U-shaped curve. Yeah, so if there's a regression method that can find like exactly that shape curve, like even a parabola, so a square regression might even work, not just a linear one. So that could be one. The other thought that strikes me is if you separate them out, you already mentioned that, into two clusters, whether by classification or whether by clustering, and then just perform two separate regressions on them. And for everyone else who's listening, these kinds of sort of combined methods where you chain together different methods together are really, really common in the real world in data science applications. You look at the data, you see, oh, there are these two separate classes, so let me create two separate models for each. Then when I get a new point that I have to predict, I'll first figure out which class it belongs to and then use the model for that class. That kind of stuff is really common in real world data. So this is a great example of how a real world data set might need slightly more complex.
Okay,
all right, more
great.
Questions, people?
No questions. I'll jump in with the comment. I mentioned something about outliers. Well, here's an outlier here. We got another one out here. But look, this outlier is on the RMSD equals 0. And what I did here was a subset to pull out the ones where the RMSD equals 0 to make sure it wasn't a computational artifact.
Right.
And I didn't see anything weird in there. So that tells me that, hey, we do not want to throw this one and this one, and there are a few more down here on the RMSD equals 0 line. We do not want to throw that out. We want to take a better look at it because that's where we were dead on the money. There was zero difference. There was zero difference between what we predicted and what the actual was in these particular samples. So one other thing we can do is go back in, subset it again like I did originally, and try to see if there's anything in particular that made the difference between the two so small. And that brings up another point. Sometimes you don't want to throw out the outliers. If you're doing a statistical analysis, for example, if you're in the finance field and you're looking at the security market line where you've got return on investment and risk, and you see something that's an outlier and it's way above the line, well, do you want to throw that out? Well, maybe for your model, but you want to come back and take a look at it because, hey, it's got the same risk as all the others, but it's paying 2 or 3 times the return. Maybe that deserves a little extra attention. So this is one of those cases here where something's out there at the edge, if not an outright outlier, but we want to examine it a little bit more closely.
That's wonderfully put, Stan. I think you're perfectly right. We talk about outliers and how outliers affect the models that we create. It's important to remove them for your modeling, but as Stan said, it's important to also do it in a conscious way. Like, okay, I'm throwing this data out, but what does it actually mean, and why is it actually important? So on that note, I think we are almost out of time. And so thank you very much, Stan. And I really look forward to actually seeing how you continue that work. So keep us posted on that if you come up, if you do more analysis and more in-depth examination. This is a really, really fascinating project.
Okay, thank you. I'll definitely do that. I do have plans to do more.
Yeah. All right, everyone. Have a great rest of your day. Bye.
Okay, bye, everyone.